So the next energy system we'll consider is glycolysis. And in this case, it's going to be anaerobic glycolysis. Anaerobic being without the use of oxygen. So glycolysis is a fairly complex process. This is a very, very simplified diagram showing us a starting point, a key sort of midpoint, and the end point. In total, we've actually got 10 key reactions. Our starting product turns into another, 10 times until eventually we get to pyruvate, which is our final product. So glycolysis is the breakdown of glucose or glycogen, forms of carbohydrate. So that's where the energy is going to come from this time to phosphorylate ADP into ATP. So we can start with glucose, which we find floating around in the blood, or we can start with glycogen, which is stored in the muscles. As carbohydrates, they are compounds containing carbon, hydrogen, and oxygen. So glucose, as you see at the top there, C6H12O6. We need to turn those products into glucose 6-phosphate for the system to work. If we're using glucose, we need to use some energy from ATP to do that. If glycogen, there's no need. So we're now in a deficit of 1 ATP. Glucose 6-phosphate then goes through a number of reactions, and we are left with 2 molecules of pyruvate, C3H3O3. So what we've managed to do there is remove some hydrogen, and that's what's going to give us the energy we need to make these 4 adenosine triphosphates. As part of the process, you can see there we use 1 more adenosine triphosphate. We then remove a number of hydrogens, and that removal of hydrogen involves breaking a bond, the release of energy, which then gives us ATP. Really, really important there is the fate of these hydrogens. We have a molecule, like a shuttle molecule. We call it like a shuttle bus. It has to pick the molecule up and take it away. But because we've only got a limited number of NADs, it's got to drop it off somewhere before it can come back and get more. So NAD picks up a hydrogen to become NADH. That is a reaction that we call reduction. Okay, reduction is the gain of an electron. So NADH plus H is NADH. NAD is reduced to NADH. If we had the opposite reaction, losing an electron, that becomes oxidation. Oxidation is loss. So when NADH is added to oxygen, it is oxidized to NAD, whereas the oxygen picks up the hydrogen and becomes H2O. So oxidation is loss, reduction is gain. Oil rig. Now, as we kind of just alluded to there in that example, in the ideal world, we attach those hydrogens to oxygen and they become water, which we can get rid of. If we don't have oxygen available, such as this anaerobic exercise we're doing, high intensity work, that can't happen. So that hydrogen has to go somewhere else. So key to that is our production of lactate. Pyruvate plus hydrogen equals lactate. So NADH will give its hydrogen directly to pyruvate. That becomes lactate, and NAD can go back and pick up more hydrogen, allowing energy production to carry on. This is catalyzed by an enzyme called lactate dehydrogenase. So the production of lactate is a really, really key reaction that allows us to carry on exercise at a high intensity. The downside is that once that lactate gets shuttled out into the bloodstream, it dissociates into lactic acid and into hydrogen, and hydrogen causes fatigue. As hydrogen builds up in the blood becomes, and in the tissues even, in the muscle, it becomes more acidic, so pH drops. And that pH triggers pain receptors. That's why we start to hurt during high intensity exercise. It is hypothesized to interfere with cross-bridge formation in the muscles, so directly stopping the muscles contracting, and it definitely interferes with the activities of some of the key enzymes, so that therefore slows energy production down and causes fatigue. on that note of key enzymes, the process of glycolysis, like we said, that's the more complicated view with lots and lots of reactions going on, each one catalyzed by its own key enzyme. The really important ones we need to consider, first of all, phosphofructokinase, or PFK. That's what we call the rate limiting enzyme. So that's occurring at that stage where we're becoming fructose 1,6-biphosphate. Now, this is the key area where we think hydrogen slows down that action, so glycolysis stops. And there's a nice safety mechanism, so we don't become too acidic, really, and cause serious damage to the body. Lactate then, and the hydrogen that's produced, can be processed, but again, we need to be at a lower intensity. We need to have oxygen available so we can break those substances down. Glycolysis can power exercise for maybe 60 up to 120 seconds, which keeps us working at a very, very high intensity exercise. But anyone who's tried to perform maximal exercise for that duration will know it is extremely uncomfortable, extremely miserable, and quite painful. So we need to then rest for an extended period of time to allow those metabolic byproducts to get processed and out of the system.